to do it in time, just to be sure that you will have some space for the question. And my lectures today is actually not even lecture, it's some sort of journey, how we can come from next generation sequencing assay, which is quite known today, and you know, each one is using, but how we can do this assay, how we can use this assay pragmatically to estimate human microbiome. And I would like also share with you first data of first Ukrainian atherosclerosis cohort. I'm very proud to share this data with you because it is very difficult work, I would say hard work with the patients and with the healthy population, because probably you already know that atherosclerosis, it's something what can affect your you know, vessels without or with different type of disease, and it's like a silent killer who can make different problems to us. And this is a first examples also of successful clinical exploitation of not only next generation sequencing technique, but also of all omics-based data medicine. And I will be probably have a chance to very briefly to tell some connection between microbiome and 3P medicine and who is actually YEPMA people. Also a few words about methodology. And methodology, I'm not going to describe what you already know during the school, but rather I will be focusing on comparative assessment of different type of methodologies. Of course, we need to at least to understand how microbiome connected to health. And the next will be providing some real implementation of next generation sequencing and microbiome data by others teams who are doing in the world. And there are a lot of companies now dealing with this subject. And also when we are speaking, as I mentioned, about experiments, it's nice, but when we are come to a real clinical world, we need to think about how sensitive it is, how this assay, each one is sensitive or not, and how we can come to from some data to real biomarkers. And I'm happy to share with you also some new determination about humans. We are all cannabinoid people or eco-holobiont or some other names is also applicable for us. So we are people who are connected to environment. And of course, environment are changing us a lot. And there are a few slides will be about cardiovascular disease models and the results which we obtained yearly, not now, recently, not within this cohort study, and we use them as background. And when we start to run atherosclerosis cohort, we're happy to report that we already done 100 plus more people. We have a unique microbiome data for our Ukrainians citizens, and this data connected to biochemistry data, immune response data, and most important issue to endothelial health data. That means if we are will be good enough, and I will share you that probably I will prove that we are good enough, that we can come to via a different type of mathematical, not statistical, but mathematical prognostic models, like, for example, principal component assay, we can really lead you to the prognostic model. And then having developed risk models, IT tool, and also algorithm, we're really happy that probably we will have some decision, and in real medicine, it will be used for teaching, for prevention and treatment of different type of disease, infection and non-infectious disease. Actually, when we are speaking about human microbiome and 3PM medicine, human microbiome, you know perfectly what it is. It's some sort of collection of genes of microbiota which covered our body on different sides. But when we are going to speak about 3PM subject, it's medicine of future, but it's not future. It's already implemented today. It's next stage of evidence-based medicine. It's medicine preventive, predictive, personalized, and patient-oriented. That's why patient-oriented, and people will tell P4 medicine, 7P medicine. And of course, it is true, but the main issue is, or it can be omics medicine, can be even quantitative medicine. But no matter how we call it, we should understand that these are two issues connected because both of these issues, human microbiome, CPM medicine, very individual, both of them are managing our health. Both of them very complex issue, and both of them very strongly dependent of background what we have. We cannot take them separately, and we need to understand whatever happened with us, it's a result of a huge interplaying cross-reactive, cross-talking mechanism, which has actually determined not only our condition, but what is most important, healing process. And there's some results which you can find in the literature. I even did not put a reference because it is, I guess, everywhere. There are a lot of data published nowadays that each typical healthy person, of course, inhabited with trillions of different microbes. And what we really know that all these microbes, they are located everywhere, and actually they are very strongly interact with our own cells. And most important issue that two healthy person can have a different microbial communities and still be healthy, but the same is true for a diseased people. And actually, when you very familiar with microbiome data, you can even predict whether this person will breastfeed or even is it educated in Cambridge or in Odessa National University. So if you're very good in microbiome data interpretation. Most important issue in this slide that you can really use different type of community to predict, first of all, them for one person for the same person based on saliva data. You can predict the gut community, and also you can use this data for a treatment, and you can predict a different type of disease. But key issue is predict. So to predict, to know how to use it, we need really to be able to create microbiome profile of the patient. Let's say it's not, what means patients? It can be created for different type of people, because we are not patients, we are actually population which are quite suffering from different type of environment effects. And the idea how to use the personalized microbiome to maintain our personal health. And there are a lot of challenges on this way. First of all, I will underline only two of them. First of all, it's very difficult to track daily changes, not daily, it's months changes. Microbiome is also changes, subject of changes. And another issue, it's very different and difficult to tell what means normal, what means regular for individual person composition of microbes. To present in just in 2 slides what means YEPMA and what means 3PM medicine, I would like to invite you to read the paper or to read the whole YEPMA journal. It's actually open access journal, and you can find a lot of information there. But the white paper about European Association for Predictive, Preventive and Personalized Medicine was presented in 2012 in Brussels. It was a huge meeting, and it was just separated countries who initiated. And now I'm so happy to report that YEPMA is also come to Ukraine, or in other words, Ukraine come to YEPMA, and our university is institutional member of YEPMA. But what is actually the difference between, let's say, classical medicine and preventive medicine, 3PM medicine? It's actually a different approach, not disease care, it's optimal health care. So we change a paradigm completely. Here we have some sort of reactive medicine. You know that nobody really in reactive medicine, even it's evidence-based and based on protocol, can really predict what will be next after your treatment. And the optimal healthcare, meaning stop disease here. So it is individual patient profile, which I already mentioned, and it's predictive diagnostic. So it's early diagnostic. It's also very targeted prevention, which is stopping disease here, and preventing different type of comorbidities or different complications. So you actually can finally treat the patients as a whole complete person, not to treat different type of organs or system. Methodology, it's a next part of my slides and today lecture, which I would like to share with you. But methodology, I'm sure you very well already know, because I'm sure that a lot of speakers already demonstrated methodology itself. But I need to tell it because it's very important to understand that whatever your experiment is doing, your methodology, if you are speaking about next generation sequencing, dependent of all these different processes, okay? RNA extractions, then purification process, and fragmentations, and of course you need to apply reverse transcriptase to get from RNA DNA. But the most important will be next steps, especially last one. When you have all this adaptive ligation, clone amplification, and sequencing itself, you're just getting very, I would say, gray raw data, which you really cannot proper explain or even use, especially in medicine, if you are not applying very properly bioinformatics approach. And then only you can come to some sort of biological insight, which you can use if you have some additional instruments. So here you can see how PCR coming with some metagenomic DNA. When we have Illumina, for example, procedure and sequencing model, then we need to understand that, okay, we will get some taxonomy, of course, we can make a conclusion based on a normalization procedure you can see here, so some normalization procedure when you can use a different type of instruments like QIMI, LETC, which is allowing us to detect alpha diversity, beta diversity, but also here is very important predictive functional profiling. There are different type of program which is allowing you. So these programs are available, and only this one led actually to understanding is biomarkers or not. Even I would be happy to share with you our approach, which is developed by us, and that specific approach, which is led to interpretation of data which we got in our cohort. So of course, this is a procedure I guess you clearly know. So it just polymerize how it's happened. I will not take your time here, but I would like to demonstrate you some paper where you can find a lot of about the program QIMI too, which really enables comprehensive end-to-end assay of diverse microbiome data. And this approach is fantastic because it's using all the data which is publicly available. So you can use any type of data and use this approach to examine them. When we comparing, this is a part about comparison, different type of examining microbiota and microbiome. When we start to think that microbiota is not needed to be examined anymore, we are not right, because we still need to examine culture methodology, and I will prove you with some new data that it's very important now. Just few words, we know that culture methodology allow us to deal, to play with live microorganisms. So we can actually using different type of test to think how we can restore microbiota. Of course, it's very quick. It's not taking 2, 3 or 4 weeks, but the main disadvantages, you know, we cannot really culture uncultured bacteria, okay? And when we come to real-time PCR, it's look like much more better because it's allowing you to have a conclusion because about uncultured bacteria or microorganisms. But the issue is you cannot really find those for which you don't have a primers. And again, you cannot even understand which organisms from them alive or not alive. You cannot manipulate, you cannot play with them. When we come to 16 RNA sequencing, it's much more better, of course. But still, it is not providing information, is these microbes alive or not alive? And here we have another troubling. We have, instead of numbers, we have a ratio of different type of microorganisms. Of course, we cannot tell even simple words about their function, okay? And next generation sequencing technique is really much more better. It's providing not only different type of sequencing material, I mean, bacteria like this microbiome, but it's also composition itself, but it's also can lead to function and to functional properties of different composition of microbiome. You can see here some real examples which is used already in medicine. So it's data not from us, but it's used for my zoo. The company is doing a very, very fantastic profile, and why it's important, because you can see here also different type of species. But as I mentioned, there are some problems with percentages. And when we come to whole sequencing, it's everything now you can do with complete sequencing, shotgun sequencing. But the issue is, of course, we can determine pathogenicity, we can know everything actually, but the issue is it's quite expensive methodology, so it cannot be applied now in medicine. And there are still some limitation. There are still, you cannot find DNA, RNA, based on DNA, RNA technique, which microbiota represented is alive, which is not alive. And again, we have only ratio of microbes. So the best solution will be, I'm just showing you 2 slides, but I need to tell you that it's probably our future, even it's not future everywhere. So the best solution will be to not detect microbiome at all, just to come to some sort of dactyloscopy, if I can say. So in each organ, we have some sort of what is left after microbiome. And it's really decision for early diagnostic. It's monitoring of treatment results like this. But it's a detection of metabolites which are connected to different composition of microbes. Why I'm saying this is a future, because at least in our lab, we now currently cannot use this methodology. And of course, the most important will be to have some sort of translation. We have translation medicine already, but you can read this paper. Actually, it is about new paradigms in public health service, an integrative approach, how we can really translate from genome to proteome. And also it's the same for metabolome. And this is actually connected also to microbiome detection. And I guess it's a very near future, because I will show you a few slides when and who is started to do it. So you can see here that I will not repeat, because this table is actually summarizing difference between metagenomic. Now we are speaking not about next generation sequencing, but next step, metagenomic and culturomic. And you know that, okay, we can keep criticize culturomic methodology, but I should tell you that there are some new technique. It's Malditov, and you probably heard a lot about this technique, and it's comparable, at least sequencing 16 sRNA application, or even with some next generation sequencing data, because the team which is led by Gilbert Greb, who is working in Institute of Pasteur in France, he is involved in human microbiome project. Actually, his team are dealing with how to compare all the different techniques which are already available, but mainly, using the best technique benefits to translate them to Malditov techniques. So, of course, we have here some, nice results, because 200 species can be detected. But when it will be developed, I believe that during this table, we have be able very soon probably to use Malditov technique just in full automatic laboratory to have the same data, if it's automated, which is connected to or connected or comparable with sequencing data. This is something new what is known. So the next part of presentation will be why it's important, but I'm sure you already know this, so it will not take long. You know how much publication now about human microbiome and health connection. So it's supposed to be fast. You know now that human microbiome is actually maintained all different stages, okay? And even more, different type of dysbiosis, Clostridium difficile infection, autoimmune disease, different type of disease. And why it's happened, how we can actually distinguish between, let's say, bad microbiota and good microbiota. So to make my story very short, I need to tell you that it's actually cannot be even mentioned this way. We do not have, we have, of course, commensal bacteria versus pathogenic bacteria and the immunity, but all the trigger specific molecular connection with tissue, everything here investigated very heavily after 2007, we come to conclusions there are no good and no bad bacteria. So you can actually find this paper by yourself. And why it is like this? Because when we see, for example, let's say what has happened in when it's happened in tissue, lean and obese patients, we can see a huge difference. Even it's not some real disease happen, but completely it will be completely different regulation. From IL-10 and IL-17, we have a different response, not T regulatory cells. Actually, we have a Th1 cells responding, and instead of, let's say, balance, we have a real inflammation. And the issue is when we start to see which type of bacteria doing this, we understand that, as I mentioned, the problem is for a different individual, different bacteria will act differently. So we cannot tell that exactly this bacteria or this diversity causes the disease and another will not cause disease. Why? Because and also we need to take into account that it will be completely different in different stages. For example, when we have adult organism or we have a child, we have something in similar. That's why in all the publications we have Akkermans, we have a company, probably you know it's company Akkermans created, which is called like next generation probiotic proposed. Or, it probably can be rhinococcus coming very soon. But based on individual profile, you cannot really solve this disease because you have a complex picture, different even from age, but also it will be based on individual differences. For example, this study was clearly demonstrated that same lactobacillus strain, exactly the same. When it's act in health person, it will be promoting weight gain. And if it's applied to obese person, it will lead, it may reduce weight gain. So it is the same strain, but implemented in different hosts will act differently. So the conclusion here that we need to have a really different profile each person, and it's already used by, for example, different team from Israel team. For example, they create some sort of algorithm based on microbiome detection, blood test, questionnaire, anthropogenic characteristic, food diary, and they are really making personalized nutrition. Only probably, of course, this all assay is supposed to be done to come to the person, and it's quite easy because all this connection based on glycemic index response, different individuals to different food, you can see here. But probably the and it's very good data because it is you can see here with some data which is connected. So if you have all this data as one panel, so you can see only the black dots, the data is not good, but all the rest data is connected. So it is proved by principal component assay that this data is connected. So we have a strong correlation which is proving that it can be applied. And it's applied for a people. You can find this day 2 instrument tool, it's available. But the issue is only that it is not really good for everybody. Why? Because it's only for obesity people, but you not really can predict to different type of disease. You cannot use this tool. And another example, very great example, this is fecal transplantation. It's also technique which can allow you to change your microbiota. And for this, you actually need professional personalized profile. But if you will read this consequences report, you will find that unfortunately, nobody is really doing next generation sequencing. Now I will come to the final part of my presentation, actually what we are doing. So I guess it will be not surprising for you that we are combining all the methodology, culture and not culture technique. Also, we have a very nice, quite expensive machines which are confirming that we isolate DNA very pure, best condition. Of course, to get this atherosclerosis data, I would like to actually show Odessa Professor Volodymyr Artyomenko, who helped us a lot with providing data from Odessa. I would like to tell that this cohort study covering all Ukraine, not only Uzhhorod. So there are different regions: Kyiv, Odessa, Transcarpathian region, and one more, which is data and processing it from east of Ukraine. And of course, it's very important. It was very difficult work to collect different samples, transfer, but it's done. And here I come, as I mentioned, to some background, which is published, so you can find it. But why we decided that it's doable? Because we found done a lot of work with the detection obese people and people with type 2 diabetes, also separately atherosclerosis, cardiovascular disease, without this next generation sequencing. We done a lot of study before, and this study allowed us to find actually real differences when you can observe in different groups, let's say in different groups of microorganisms. So here we have enterobacteria group, and before you can observe Kotzi, and here we also detect different type of Candida. Even I should tell you that Candida and also Bifidobacterium lactobacillus and Clostridia group. I went from IPC Congress. It was very interesting observation that microbiome can completely change our let's say our view about what we really need to put in individual microbiome profile. And also it is different, it's very important because it's anaerobic, different representatives of anaerobic bacteria. It's very important, but as I mentioned, you can read this paper, all this data provided, this work done by Tamara Maleshko. And I'm so happy that finally we come to some sort of summarizing data when you can really see which is difference, which is the similarity and difference between the different type of microbiota representatives, because as I mentioned, it was not next generation sequencing data. So we are actually based on this when we come to the project. And in that time, we had really difficulties how to tell what exactly we can call biomarkers, because this is nice picture which I just showed to you. It is, of course, showing some differences, but to be very clear, what you can really treat as biomarkers, we use a completely new approach, which nobody used before, I'm sure. And it's actually also published, but our idea is to use not statistical, not statistical biomarkers, but dynamic biomarkers. Finally, we came to the exact line what can demonstrate us that from 62 different type of indicators, only 12 we can really treat as biomarkers. And then we decided to start our study, which is project supported by Ministry of Education and Science. It was a 3-year project. It's still running. This is last year. And we, of course, check a lot of already published material. We were oriented on sequencing not only microbiota, but also functional profile. And as I mentioned, since we are dependent a lot from a different influence from nature, like exercises, diet, sunlight, let's say our lifestyle, we created quite complicated study, which united everything was based on what was already known. It's our work together with a Slovak partner. So we already know some prediction which markers probably will be very informative. And we also focused on completely new machines. It's a name machine atherosclerosis TL2, allowing to detect real markers. What I mean real central systolic blood pressure, aortic pulse, wave velocity, different techniques which are really like very strong medical diagnostic, which we connected with the microbiome data. And also quite similar things were measured: peripheral blood pressure, indicators of systolic, diastolic blood pressure, etc., etc., etc. So it was very strong medical data. And this medical data we also tested before with the Slovak team. We published a paper about stroke stop formula. So what it's supposed to be do, but it was not with microbiota study. It was separately based on exactly on clinical study when the symptomatic and you can see asymptomatic people were analyzed. And it was some sort of conclusion before we started the project that there are 3 types of plaque: atherosclerotic plaque, mixed, soft, and hard. And based on this, actually, there are some sort of algorithm and formula was proposed. And we use all this as a background because it was detection correlation were detect, and we based on this correlation we choose prominent biomarkers, and this is data which we obtained, a different study I would like to share with you. So you can see it's real cohort. I just cannot show you all the 100, of course, persons. And we have a group, control group, and experimental group, and we measure all these physical features. So it was the index and the height, weight, BMI index, waist measurements. There are a lot of physical features. You can see that there are no sometimes very huge difference, and this is actually the benefits of cohort, because you have real people, let's say people from the street, some of them obese, it was some sort of inclusive exclusive criteria, so no of them had a different type of disease, but it was strong stratification procedure for people with obesity, without obesity, people with diabetes type 2 disease, without the diabetes type 2 disease. And this table are actually showing biochemistry. So we have for different type of, let's say, control group and experimental group, different type of low density lipids, high density lipids, creatinine, so homocysteine, and few measurements. So, and after that, we, of course, come to the sequencing part, which I actually would like to share with you so much. So our median frequency per sample was 338, and the median frequency per feature was 4,500. It was around 400 different identified genes, and the deep was like more than 20% uncultured and cannot be found even by any type of routine methodology. And 83% of raw material were identified to genes and 70% to the species level. So I'm really happy to share with you this data. You can see clearly a difference between different groups. Mainly, I will make a conclusion, so you will see it. So in Fusobacterium, we have observed the difference. It also actually a huge difference in Myxococcota and some other groups which were not really reported before. But mainly it's Actinobacteria and Fusobacteria. You can see the difference, and you can see also the huge difference in I'm trying to put it separately, but I hope you will see it later, where the presentation will be shared. That also on family level, not on the species level also, but you can see clearly there are so much data. I will not be since the time to tell everything, but you can see relative abundance indices is very, very characteristical for experimental and control people. And let's come to the individual patient microbiome profile. So you can see how it's look like. It's here you have in percentages, but it's also available an exact number. And you can see how different it is for each person. And mainly you can see it's much more that it was done by different type of companies. And we are happy that all this data now in examination for a specific risk model. And what we observed, biodiversity in control group is generally higher, but it was something expected. Relative abundance of oscillospiraceae and, as I mentioned, Fusicatenibacter, Erysilotrichaceae, other species like Stensenelaceae group, Monoglobus, Moribaculaceae, Coriobaterias, Anaeroviraceae, and some other group is higher, much more higher than in control. But we are happy to report that such a group like Veilonelaceae and Moribacteriaceae, Archaea, Succivibrio, Microbiome, Akkermansia, of course, bacteroides, which were found in control, but they were not found in an experimental group. But we are happy to report it because we observed not only typical representatives like usually reported, Akkermansia bacteroides, and look like not Ruminococcus is species are really, or species or genera is not really diagnostics, have any type of diagnostical value for a Ukrainian cohort. And now probably the final part, what is actually we can do with this data in medicine, because we are speaking about medicine. So if you see this data on a different scale, not like scientific data, we can easily tell, for example, how we can explain what has happened. For example, it's one person, let's say any type of person data you can observe, and we can tell that, okay, here we have a dysbiosis. Why? Because it's some problem with the protective species and another problem with the histamine specific bacteria, histamine producing bacteria. So we can come to the conclusion that it's, let's say, dysbiosis. But then when you probably remember enterotype, different characteristic, we understand that there are different people enterotype exist. But here, when you come to individual data, you will be clearly see that it's not detected enterotype. Enterotype based on mainly Bacteroides prevotella ruminococcus species, so we can tell that it's individual specific. And here, based on this examination, we can clearly tell butyrate predation, but clearly detect butyrate, lactate production, bacteria degradation, mucin degradation. So we can see here, so it's some sort of specific formula for any individual, which can, such characteristics, Shannon index, Firmicutes bacteroides, ratio, butyrate product, lactate, acetate, as I mentioned, mucin degradate, LPS, Prevotella, different type of species. And you can make some conclusion about neuroactive microbiota, about functional bacteria, about protective microbiota. And this all is leading finally for also metabolism profile detection. And in connection with function and with exact profile, we can come, for example, for some sort of detection of different type of disease or risk parameters. For example, here in such person, we detect allergy, asthma risk parameters. But of course, the same way you can detect IBD, atherosclerosis, cardiovascular disease, and different type of disease. So this is the issue. How to do it, and how to do it in practical terms. In practical terms, coming back that you need to have some sort of instrument which can really help doctors and patients at least to understand what happened with them, we need to have some IT instrument. And this was created in our lab, but together with some business company, ODiN, which is actually where our old people working who are working in the same center. And this is actually a lot of databases which we already created even before. And this is a database is now dramatically improved because this is project. And it's look like this. So each one can have access online, can put their data. And finally, we provide also data for gut or skin, whatever type of microbiota. And I would like to demonstrate you one of the example, which is databases created by our Roman Rukavchuk. He is going to have a defense very soon. Actually, all the bioinformatic assay were done by him. So we create a lot of our, let's say, own databases. And now we need to almost finish that if we will be able to, based on this microbiome profile, prescribe people not only tablets, but also different type of food, which is quite good. We established this food in another project. It's Vitrina Zakarpattia, so you can find it based on sites that we detect a lot of Transcarpathian food, and we prove that this food based on different type of microelements, macroelements, and flavonoids, fiber can be really good for people. So we hope that all this microbiota data and the next generation sequencing data will be useful even to treat our people, if I can say treat or prevent disease based on traditional food. And I would like to invite you to join the SAMET Society. I need to mention this because now I'm president of SAMET Society, and it's a society which is actually learning microbial ecology and disease, and I hope you will join it. And at the end of my talk, I really would like to thank you a lot for the people who done all this work together with us. It's Taras Shende was responsible for clinical protocols and also participated on the cohort study. Svetlana, who done a lot of work, Alexandra, Tamara Meleshko. Alexandra was responsible for an immune response, Tamara Meleshko for a lot of other data for leading. She was actually executor of this project. Lesia, you know, all the new people in our lab, Vlad, Maria, Maxim, but also Roman Rukavchuk and IT people who are actually creating this algorithm. Thank you for your attention.
Thank you a lot for your interesting and very full of content lecture. Dear participants, you can ask questions, you can write them in chat or raise your hand in app. So we have the first question.
Yes, I can see it, how our microbiome can affect on, I guess, on brain. And I did not cover this. Actually, I try to be as fast as I can. Probably you can see it. I even finished something like, you know, 15 more minutes because I guess...
No. It was 45. It's totally okay. It's
exactly. So I'm sorry if not, because I really cannot predict time. I was trying to be in time, but I did not cover this data. And the question is, I cannot see who exactly asked question. Probably I will ask. I will open. Yes, now I can see. Dmitry Alexandrovich, know how our microbiome can affect on our brain or our mental condition. So this is a fantastic question. Is actually what we are now analyzing, and now that how it's the answer is HABA. Okay, so it's connected via HABA, and the mechanism is already known. There are a whole group of people who are working in Dublin in the center IPC. So it's Alimentarium Probiotic Center, which is dealing with all the mechanism how our food via HABA regulate our mental health. But what I would like to tell, what we observed in our study, that not only in study, in the patients, when they have a distress syndrome with their gut, for example, it can be IBD or different type of immune disease, so they are suffering with the gut pain, they are need also additional medication. So it's not very easy only treat to microbiome, but you also need to treat, let's say, with some sedative effect, person behavior. So microbiome to respond exactly directly on our brain. Probably, in real life, when you have a not fantastic mood, you simply start to eat something sweet. And this is actually why we are, why we are using this as a HABA metabolites, improving our mood. There are some exactly connected bacteria and microbes which are connected with mental health. This is groups of bacteria which I cannot now describe exactly because we don't have probably enough time for this. But what I really would like to mention is that there are some specific, even patented now, neuromediators who can improve mental health by improving microbiome, by changing microbiome. And you simply need to come to visit IPC center. And this work, which I forgot his family name, but we met recently in IPC Probiotic Conference, Clark. He is dealing with mental health and with the connection with gut microbiota. It was not exactly our aim to study, but we already come to conclusion that it can be the very good key for atherosclerosis cohort to detect it also for some sort of indicators of brain function. Then we probably can even predict more, not only prevention of cardiovascular diseases was the aim, but also different type of brain disease, so-called brain-connected disease, like atherosclerosis, like Alzheimer's disease and Parkinson's disease and others. Another question is here from Mikhail. Yes, and so what other project not especially related to immunology are you planning to make in the nearest future? And what method will help you to achieve successful results of them? This is very interesting question, actually. We are writing right now proposal. It's and especially mentioned, not especially related. Okay, so it is a project not related to immunology. It little bit probably. It's a project we are writing right now, our team. We involved in a proposal preparation. It's Horizon Europe, ICARO. It's about how to change behavior of pregnant women. It's actually connected to the first question. How to change behavior, nutritional behavior of pregnant women, make them more quiet and happy, and prepare for them specific personalized nutrition plan, which will allow these moms being pregnant, deliver baby, which I hope will be them, since they will be very well treated, I mean, with mother care. And they will be actually yearly, it is the strategy of yearly education of these mothers who will be supported with individual nutrition plan. And we are going to do this study with EuroCut. So we will doing our sequencing. They will do all their cohort. It is a 7 pilots plan. And the issue of this new project to save children, to deliver healthy children, and to prevent in early life stage development of different type of disease, IBD, type 2 diabetes, but mainly type 2 diabetes and obesity and cardiovascular disease, since it's our topic. And another project is more social project. We are working now with Slovakian team. We would like to create a rehabilitation center, since our group working also about post-COVID rehabilitation. And now, as you probably know, we have all in country problem with post-stress syndrome, because, our, all the refugee people who are coming in our region, it's very, very strongly affected people with the war issue. So the Lekarska or whatever, Medical Doctor Association of Slovakia, together with our university, now working on the proposal, probably it will be submitted on September, which is aimed to provide real people with real support based on microbiome data also, but in other, all of the other instruments to make them really much more healthy and happy, because the people are really suffering from this. This is about projects. It's only 2 of them, which I mentioned. We are really working currently. We win first stage for Horizon Europe, and we are coming to this second stage level. And I have another issue from Ernest Heleta. What's the main factor? What's the main factor which induce diabetes? The main factor is, thank you so much, Ernest, for this is a great question. Because wherever we are speaking about microbiota, microbiome, I don't know, biochemistry, all these, let's say, biomarkers, they become only in this case biomarkers if we can prove they really, how to say, lead to disease. So the answer will be the main the main factor which induce diabetes is low-grade inflammation for individual person, which is stop or change a balance between commensal and, let's say, pathogenic bacteria, which I told that there are no good and bad bacteria. So it's individual balance changes which lead to not ability to digest properly different, not only biological active compound, let's say different food. So then we have after we have changed microbiome condition, which is lead to chronic inflammation, we have changed completely T regulatory cells activation, and it's lead to Th1 response when the organism, our host, start to produce enormous amount of interferon gamma instead of producing IL-10, and also it's produced a lot of not only interferon gamma but also IL-17. In a normal host, IL-17 is good. It's actually the factor who is prevent autoimmune diseases. But in inflamed host, it becomes the trigger to start this disease and type 2 diabetes disease. So if we can really maintain this balance individually, based on a microbiota profile, we really can prevent development of type 2 diabetes. And probably you know that it's not only in obese people you can detect diabetes type 2. Next question, first project sounds really interesting, while second looks like more actual. Good luck. So thank you. It's not a question, it's a note. Thank you for your interest. Is this a relation, it's a question from Daria Zatinaska, is there a relationship between the microbiome and mental health, and are vulnerable population differently affected? This is exactly a fantastic question. Yes, it is, because to be quite short to answer this, there are clearly reported data when we have acute stress and when you have chronic stress, it will be completely different composition of microbiome and will be completely different response. When, for example, we have acute stress, we don't have a really so dramatical changes in microbiome, but if it's a vulnerable population differently affected with different type of stress, it's completely different approach. So the first of all, it can be clearly defected on different, how to say, biodiversity. It's become very altered profile, and you can really easy, even sometimes probably you don't need shotgun sequencing to deal with these disorders. And there are very interesting for us project which we would like participate, but we did not even start to think how to do it. It's connected to work people for a stress situation here. It will be not very interesting. It will be very useful to use the data which we already know to start to produce some sort of neurobiotics which can regulate, and it cannot be only bacteria. I guess it should be some sort of composed. I don't want to use a word drug because it's not supposed to be drug. It's supposed to be, let's say, biotic, which can regulate also metabolically. So we can use, let's say, clear butyrate or some other metabolites to normalize the response. Because if you are using ready to use metabolites, it can be much more, it can be done much more faster. And yes, it is an exact relationship, and it is known already for investigation. But as I mentioned, our center is dealing mainly with real medicine, with real people. So we will be happy to come to do this for real people. I mean, for especially in our region, because our region is full of refugee people, and we are really sorry for them. And we are making communication with people in needs and some other organization who probably can help us to create this rather social project, as I mentioned, not a theoretical project, not a fundamental project like this. In Zhunko, I guess, many children with autism have changes in the gut microbiota. How much does influence the course of the disease? Could this be one of the causes? Yes, it is. This is actually our covering ICARO project. So we will not deal only with, let's say, healthy mothers, but also, of course, we will deal with healthy mothers. But we have specific cohort for children with diagnosed autism. So it will be not about pregnant, it will be separate cohort, which we would like to examine during this project. I hope it will be, we will be able to do. It's not in Ukraine, I mean, it's not Ukrainian cohort, it's supposed to be Slovakian cohort of children. But of course, I guess we can try extrapolate this data to Ukraine. Why I am quite skeptical about extrapolation, because the comment to this different, why we have actually 7 pilot, not one pilot, why we do need to do 7? Because as you know, that microbiome is a very sensitive issue, and it's dependent on everything. In our algorithm, we have also connection with the geographical location, with the different type of food which are consumers, so let's say people using, with the different databases. And we will not be, if we will simply do the assay for, let's say, Slovakian or the project leader of ICARO from Barcelona, if we will do it for Spanish population, and then simply trying to use this data for Ukrainian population, for Ukrainian pregnant women, for Ukrainian children with autism, or Ukrainian people under stress, it is not correct. So we really need to have our Ukrainian cohort. And actually, I would like to tell you that being a member of Scientific Council of National Foundation of Ukraine, National Research Foundation of Ukraine, I submit now, together with the working group, the documents about consolidation of our forces, what is very actual, I hope, to this school. This is consolidation of our forces aim to put together different type of funds and different institutions to start to do our Ukrainian cohorts, different type of cohorts, and also find the money, and actually not only money, but people, young people or young researchers who can continue to do it on different levels. and also combine not only our efforts, but also funds. And the Ukrainian cohorts must be, and the Ukrainian databases must be, and Ukrainian biobanks must be. And I understand that it is wartime, but I guess sometimes wartime will help us to be more pragmatical, more united, and I guess we will succeed it, because Ukrainian science will be, and it must be. So I would like to thank you, each one who joined us today, and thank you for your kind attention. And you have all rights to contact me anytime. I would be happy to communicate with you with any type of knowledges I can share with you. Thank you.
Thank you a lot. There is one question in chat, or you just have no time for?
No, I cannot see it.
It's from Inna Zunko, the last in chat.
Could this be one of the case of disease?
Many children with autism have changes in the gut microbiota. How much does it influence the course of disease, and could this be one of the causes of the disease?
But I just, this is what I just answered.
I'm sorry, I was
that yes, and you know,
I'm very sorry.
It's very altered, and it's completely unique, because, and it's proof that it is influencing on you know, behavior, and it can be improved. And actually it will be, I cannot tell more, because it is something what we are going to investigate within ICARO project, of course, if you will succeed it.
Yes.
But it's the Slovakian group, if we will not succeed, they already doing this research. And if Inna Zunko interested in exact data to like come to the species level, I will be happy to provide her when any time, you know, just send the data. Thank you so much.
Thank you for this lecture and these answers, and we have a hope to see in real life in some conferences, and maybe maybe the summer next year will be offline. So it was great to see you. So
thank you.
Thank you.
Bye.
Bye.